Hello everyone, and today I'm going to be talking about hemostasis. Note, this is hemostasis and not homeostasis. Hemostasis is just a fancy word meaning how the blood, makes sure that it stays inside of your body rather than outside of your body, which is, primarily where you want to keep your blood. So stasis means staying in one place. Hemo is your blood, and the primary way that your blood stays inside of your body is by making sure that when you get an injury that would cause it to leak out, it clots. And so this is primarily going to be about the blood clotting reactions. And the other, there's a couple more mechanisms that blood uses to stay inside. So, we've talked about a few different things inside of the blood at this point. We've talked about the cellular bodies. We talked about red blood cells extensively. We talked a bit about white blood cells. We're going to talk more about those when we cover the lymphatic system. But we haven't really talked very much about platelets. We know that platelets are not actual cells themselves, but they are cell fragments. And we know that they're somehow involved in clotting, but let's going to take a deep dive into that right now. So, platelets, what are they? Well, they come from these big cells called megakaryocytes. And when I say big cells, I am not fooling around. You can see a picture here. This is a megakaryocyte, and you can see the relative size of the red and white blood cells next to it. So this is a huge frickin' cell. But it doesn't flow through your blood. In fact, it probably couldn't flow through your blood. It's too big to fit through a capillary. But what it does do is near your capillaries, or actually in your sinusoid capillaries in your bone marrow, it sort of extrudes itself into these long tentacles that break off into fragments that eventually become platelets. While these platelets are not full cells in and of themselves, they actually do maintain many of the functions of life. They have, some metabolic factors. They can have mitochondria inside of them, so they can still make their own energy. You can make an argument that they're, better, more complete cells than red blood cells are. But they can make little pseudopods, they can engage in some phagocytosis, they've got a bunch of different functions going on, and let's talk a little bit about what the main functions of platelets are, right? So, first main function of platelets is that in the event that you suffer some sort of injury, which actually happens all the time, it isn't just when you get a cut from the external environment, your capillaries and your minor blood vessels are undergoing, small lesions and small cuts that need to be repaired on a pretty regular basis. And when that happens, platelets secrete vasoconstrictors that help reduce blood loss. We'll get into that more in a second. They can also stick together to form what are called platelet plugs. We'll get into that in more detail as well. They secrete proteins called procoagulants, or clotting factors. These are important factors for the clotting reaction, which we're going to talk a lot about. They also help to get rid of clots. Clots are, by their nature, supposed to be temporary. They're going to stop the blood loss while the healing process begins to take place. But you need to be able to get rid of a clot. Otherwise, they could stick around. They could do a lot of damage. We'll see a little bit about how that happens later on as well. In addition, platelets, since they are going to form clotting reactions, they're going to be at high densities at the site of injury, and injury is a place where infection can potentially take place. Any cut is potentially a site of infection. So platelets are also going to release chemicals that will attract neutrophils and monocytes to the area. Those are two of your white blood cells, and they're going to help clean up any mess that's in there. Neutrophils, of course, are your body's main, They're your body's army. They're your body's main defense force. Monocytes are going to be what will become macrophages and dendritic cells, and they're going to be important both for cleaning up any bacteria or viruses or anything else that managed to get in through the cut, but also because as that clot begins to break up, begins to dissolve, those phagocytic cells are going to need to basically eat the debris so that it doesn't stay around. Platelets can phagocytize and destroy bacteria. That's not, their main function, but yeah, if they encounter a bacteria, they can eat it. And last, they are going to secrete growth factors, what's called platelet-derived growth factor. This is a healing factor that tells cells, stimulates them to undergo mitosis and to divide, and that this is going to promote the healing response, so that, your cells begin to proliferate and begin to fill in the hole of whatever sort of injury you started to suffer. So, what are the mechanisms of hemostasis? What are the mechanisms by which your blood is going to stay inside of your body? We'll see that there are 3 mechanisms, and platelets are pretty intimately involved in all 3 of them. The first and fastest one is what's called the vascular spasm, right? Anytime there's an injury to an artery or a vein or anything like that, your blood vessel will undergo vasoconstriction, right? So that injured blood vessel and sort of all along its length, it will constrict, and that will make it so that less blood flows there, right? It will shunt blood away from that area, and that means that you will lose less blood. What sorts of things can trigger this vasoconstriction, this vascular spasm? Well, pain receptors, pain can cause it, and pain actually makes a very short duration spasm, but it's the fastest one, right? So pain can cause vasoconstriction. Smooth muscle injury, like the death of smooth muscle cells. Remember, smooth muscles are found in the wall of arteries and veins. Well, when those smooth muscles are injured, they die. They send a signal to all of the smooth muscles nearby that causes those smooth muscles to constrict. And last, platelets, if they detect a breakage in the blood vessel, they release serotonin. And if you'll recall from way back in the previous semester when we were talking about the autonomic nervous system, serotonin is typically a vasoconstrictor, so it would cause vasoconstriction. Generally speaking, pain is going to provide short-duration vasoconstriction because not all pain actually results in blood loss, right? But you don't want to wait for anything else. You get pain to an area, you shunt blood away, just in case. But if nothing else happens, then you can let blood flow back to that area. However, if you have pain coupled with smooth muscle injury, then that means that there was actually some damage done to the artery or done to the vein, and then you're going to have a longer-term constriction. In this case, long-term is talking like, minutes, tens of minutes, maybe an hour, but probably not hours. So even the long duration here for the vascular spasm is relatively short term. That's because you are eventually going to need blood to get to that area in order to heal it. But the whole point of the vascular spasm is to shunt blood away, and that's going to give time for the other 2 mechanisms to come into play. The second mechanism, which definitely intimately involves platelets, in fact, it's named for the platelets, this is the formation of a platelet plug. All right? So platelets, they have proteins on their surface that allow them to stick to things, right? It allows them to stick to cells, it allows them to stick to each other, but you don't want platelets to stick to things inside of your blood vessels when you aren't injured. That could be really bad. They could stick to random surfaces, and that would cause, a big bolus that might shut off blood to an important organ, like the heart. And that would, that's bad when that happens. So intact blood vessels, like blood vessels that are working properly, are coated with what's called prostacyclin. And prostacyclin stops platelets from binding to the artery or the vein, the blood vessel in general. However, when the blood vessel is injured, right, that prostacyclin coating is disrupted and the collagen underneath shows through. So when that collagen pokes through, well, platelets can and do stick like the dickens to collagen. And platelets will then stick out little pseudopods to bind to the collagen, and they'll start to plug up. You can see here this exposed collagen right there, and the platelets are sticking to that collagen. And not only that, but once they start sticking to collagen, it causes basically a positive feedback reaction where they will also put out more pseudopods and grab more platelets. And so then the platelets are going to actually stick together as well. And so they put out these pseudopods, they grab onto the collagen, they grab onto more platelets, and then they pull in the pseudopods, they contract, drawing this plug together, this really dense plug of platelets all stuck to each other. And these platelets contain granules inside of them, granules of chemicals, several different types of chemicals, one of which is serotonin, we already talked about. And, they'll be releasing that as soon as they detect that there is any collagen in the wound area, and that causes vasoconstriction. The granules also contain ADP, which is actually a platelet attractor. So it will attract more platelets to the area to make the plug bigger, and it will cause the platelets to degranulate faster. This is a part of that positive feedback loop, right? You get a few platelets binding, and then they degranulate, they secrete ADP, and then that causes more platelets to bind, and then they degranulate even more, and then they secrete more ADP, and that causes more platelets to bind. And so you get this positive feedback reaction forming this platelet plug in the localized area. They also release from their granules thromboxane A2, which promotes platelet aggregation and vasoconstriction and things like that. Right, so that's second here, right? The formation of the platelet plug. This is not as fast as vasoconstriction, as vascular spasm, but it still happens pretty quickly. This happens in the first few minutes after an injury. All right. The platelet plug will usually shut down the use of that artery, right? But the platelet plug is not a long-term structure. It will eventually degrade unless something forms to hold it into place. And that thing that's going to form to hold the platelet plug into place is what is called a blood clot, a coagulation. This is the most effective defense against bleeding, and it's also the one that takes the longest to happen. It's the last one. It's the most highly regulated. So in the coagulation reaction, we're going to talk about this extensively in a couple of slides. But basically, you've got a whole bunch of regulatory proteins that kick off, but the ultimate point of them is that they are going to convert this protein called fibrinogen, which is present at high levels in your blood, into fibrin. That fibrin will then polymerize into long chains that form a network of insoluble fibrin protein. That network will worm its way through the platelet plug, basically stabilizing the platelet plug and making it into a permanent or semi-permanent structure. What causes the the coagulation to happen, there's actually two things that can cause it. There are two what are called pathways that can activate blood clotting. There's what's called the extrinsic pathway. The extrinsic pathway is activated by tissue damage. You have a cut, you have some trauma to tissue. Those dead cells spill out this protein called tissue factor, and that's what's going to initiate the cascade. The other way that platelets can form, that blood clots can form, involves platelets, and it's called the intrinsic pathway. The intrinsic pathway is going to be a blood clotting pathway that is initiated by platelet degranulation. Right? So this thromboxane A2 and some other proteins that are released when platelets degranulate, that's going to initiate the clotting cascade. So those are two different ways that clots can form. By the way, most clotting enzymes and clotting factors, including fibrin or fibrinogen, are made in the liver, but they're present in the plasma of normal people. So, let's take a little bit of a closer look at how coagulation is going to happen. And this is going to be a kind of complicated look. So, pay attention. There's a lot of proteins going on here. All right? So we said that there was two main pathways. There's the extrinsic pathway, and there's the intrinsic pathway. Both of them result in coagulation. So both of these pathways are actually going to meet together at what's called the common pathway. So no matter which mechanism you start by, there's a common pathway that you will meet at. I'm going to start by looking at the extrinsic mechanism. It's a little bit easier to understand, right? So the extrinsic mechanism assumes some sort of injury, right? You have some sort of external factor that causes tissue damage. You fall off your bike and you scrape your knees on the pavement. You get a cut with a knife. You step on a broken branch and it pierces your skin. Something like that. Well, your tissue cells, so cells in your epithelium and in your connective tissue and whatnot, the paravascular tissues, right? So that means the tissues that are close to your blood vessels. When they are damaged, they release a protein called tissue factor, or TF. Tissue factor. And this tissue factor is also called thromboplastin and factor 3. Like all of the important blood coagulation factors are all named like factor 3, factor 9, factor 13, and whatnot. They're named in the order in which they were discovered, not in the order in which they work. So, like, it doesn't start with factor 1 and then go to factor 13. That would make too much sense. So this extrinsic mechanism is going to start with factor 3, but I generally call it tissue factor. I like it because it's released by tissues. This tissue factor, when it gets spilled out into the blood in the presence of calcium and factor 7, becomes activated. It actually activates factor 7. So this tissue factor will activate factor 7, and together they will cause factor 10 to become active. By the way, you'll often see these things referred to as, like, say, factor 10, right? It's factor 10, but FXA is the active form of factor 10. This is an enzyme cascade. If you recall back to when we were talking about signal transduction mechanisms inside of cells, you remember how we said that a receptor, you could have just one receptor that becomes activated and it releases a bunch of second messengers. And all of those second messengers are going to bind to some protein kinase, and then that protein kinase is going to become active. And if you have, like, just one thing that binds the one first messenger might lead to 100 second messengers, which might lead to, 1,000 or 10,000 activated protein kinases that are going to then activate 1,000,000 different secondary kinases that then activate, 10 million different tertiary kinases, which actually go and make the response happen. That's how you get from just one little teeny tiny thing, one little thing that binds a receptor to make this huge response. We called that signal amplification by means of a enzyme cascade. The same logic is actually going to apply here, right? So you need a big response. You need to make a blood clot. You need to make it quickly, right? But you don't necessarily have a huge, ton of injury. You've only got a little bit of tissue factor that spills in there. So at every point along this pathway, we're actually going to amplify the signal. So one activated factor 7 might potentially make hundreds of activated factor 10, right? So this tissue factor and factor 7 together are going to turn factor 10 into factor 10A, the active form of factor 10. Now, we're going to get back to this in a second, but first I want to go down here and talk about the intrinsic mechanism, right? So remember, we said that the intrinsic mechanism is activated by granules, substances, molecules in the granules that are released by platelets. Well, these granules here, they release factor 12, right? Factor 12 stimulates factor 9 to become active. So factor 12 here is going to turn factor 11 into factor 11A, right? So this is factor 11, and it's going to be turned into factor 11A, the active form. Factor 11A is going to take factor 9 and turn that into factor 9A, the active form of factor 9. In the presence of calcium, again, so this entire process depends on calcium being present in the blood, factor 9A is going to convert factor 8, VIII, right there, into factor VIIIA, factor 8A. And then factor 8A or factor 7 plus tissue factor, either of those can stimulate factor 10 to become factor 10A, the active form of factor 10. And that's the beginning of the common pathway. It doesn't matter which way you come down here, right? It all comes together at factor 10. Once factor 10 becomes active, right, factor 10A turns on something called prothrombin activator. Prothrombin is also called factor 2, but it's usually just called prothrombin. Remember how we said that anytime you see pro something, that means that it's made in an inactive form, but if you cut a little piece off of it, it becomes active, right? Prothrombin, factor 2, is made by the liver. Prothrombin activator will cleave a little piece off of that prothrombin, causing it to become thrombin. Thrombin is a protease. It cuts a little piece of protein off. There are actually several different proteins that can be cut by prothrombin, but one of them is fibrinogen, which is factor 1. Now, just like gen at the name means that it's made in an inactive form, right? So prothrombin becomes thrombin, fibrinogen becomes fibrin. Both pro and gen basically mean that they're made in this inactive form, which when modified by having a little inhibitory piece cut off will then become active. So we see thrombin here is going to turn fibrinogen into fibrin. Another thing that thrombin can do is it can activate factor 5, right? So it can turn factor 5 into factor 5A, the active form of factor 5, and what that does is it causes more prothrombin activator. So factor 3, factor 5, calcium, and PF3 all act as activators on this pathway. You'll note that this is a positive feedback loop. So you start off with a little bit of factor 10. That makes prothrombin activator, which makes thrombin, which makes factor 5, which makes more prothrombin activator, which makes more thrombin, which makes more factor 5, which makes more prothrombin activator, which makes more thrombin, which makes more factor 5. Right. This is another part of how we get signal amplification in this pathway. You have this positive feedback loop that builds on itself. Now, fibrin by itself, so fibrin is like made, so fibrinogen, right? Which I'm going to call FBG. You can think of it as these little blocks, except they've got a inhibitory domain that stops them from polymerizing, right? Thrombin is going to cut that little domain off, and it goes away. You then have your fibrin, which I'm going to call FB, and it starts to polymerize into long sections. But they're just being held together by non-permanent bonds, right? They're like Legos that are being hooked together, but you could take them apart relatively easily. They're a temporary structure. Well, factor 13, in the presence, again, of calcium, is basically going to fuse this together. Like putting some super glue on your Lego blocks, right? Now you can't take them apart anymore. You can, but it takes a lot more work to take them apart once they've been sealed with factor 8, or factor 13, sorry. So, once you've made a clot, You don't want it to stay around forever. You want a clot to form very quickly. Clotting reactions happen within 30 minutes. Sometimes much faster than that. It depends on the extent of the injury. Once the clot has formed, remember it surrounds the platelet plug. The platelet releases platelet-derived growth factor, which stimulates mitosis in the surrounding cells, which will then begin the healing process. They will divide to basically cover the area. And once the area has been covered with new cell growth and the beginning of some scar tissue and stuff like that, you don't need the blood clot anymore. So, it's important that you take the blood clot away, and it's important that you do it in a very controlled fashion. You don't want this blood clot to get loose and then just float around in your vascular system. It could get lodged somewhere and cause damage. So you want to take it apart in a very controlled way. This process of taking a blood clot apart is called dissolution. All right? So there are several pathways that cause dissolution, but ultimately they come down to plasmin. Plasmin is again a protease, and it is specifically a protease that can cut fibrin polymers and can cut them apart into little teeny tiny degradation products that can then be recycled. Basically, they can be eaten by some macrophages and absorbed by platelets and stuff like that, and then taken apart and remade. Plasmin is another one of those proteins you don't want active all the time, so it's made as an inactive precursor called plasminogen. Right? There's that gen suffix again. You've got plasminogen floating around in your blood all the time, but it only becomes active when you cut off a little piece that will, basically cause it to become active. What cuts off that piece? Well, there's actually a few things that can do so. Right? One is called tissue plasminogen activator, or TPA. This is sometimes called TPA. And it can basically cause plasminogen to convert into plasmin. It's a protease that can cut plasminogen into plasmin. This is not active all the time, right? Because if it was active all the time, you wouldn't be able to have any blood clots, right? So there is another protein called plasminogen activator inhibitor that inhibits plasminogen activator. Right? If you have too much TPA, then that means that your body is not able to form blood clots. If you have too much plasminogen activator inhibitor, what's called PAI, usually PAI1. There's a 1 and a 2, but the 1 is the one that's active in adults. If you have too much PAI1, then that means you don't make enough plasmin and you can't get rid of your clots. That can actually mean, because this is a very important system, right? We said that there's a positive feedback loop in how your clots form. Well, what is it that causes them? Because we know positive feedback loops tend to run out of control, right? They reinforce themselves over and over and over again until something stops them. What stops them? This process stops them. So you're always, anytime you make a blood clot, you're also making plasmin that's chewing away at that blood clot. And if you don't make enough plasmin, then that blood clot can actually spread throughout large portions of the body, causing your blood to coagulate. And that's bad. I mean, you want blood to coagulate at the area of an injury. What you don't want is that blood clot to positively feed back and spread throughout the entire body. So you always have plasmin eating away at the edge of it, keeping it under control. Again, there's not just TPA breaking up the clots. There's also this protein called kallikrein. And kallikrein is made from pre-kallikrein. Pre, like pro, means that it's made in an inactive form, right? So this pre-kallikrein is floating around naturally in the blood, and factor 7, which is a clotting factor, converts it into kallikrein. Remember, that's a clotting factor. It's made in the clotting pathway. So as soon as you start to form a clot, you also start to form the mechanisms that are going to keep that clot under control. Clots are absolutely essential. Without hemostasis, you suffer great injury. You bleed to death very easy. Ask any hemophiliac. It's not a fun disease to have. However, blood clots can also be dangerous. Talk to anyone who's ever had a myocardial infarction or a heart attack or a thrombosis, which we're going to talk about in just a second. If a blood clot gets out of control, it can cause massive damage to the body. So as soon as you start making a blood clot, you also start making the control mechanisms for dealing with that blood clot. One of these is kallikrein. All right, so factor 7 from the clotting factor is going to convert pre-kallikrein to kallikrein. Kallikrein can also convert plasminogen to plasmin, and plasmin, which we said was a protease, and it can digest fibrin, it can bust a clot, can also convert pre-kallikrein to kallikrein. So it also has a positive feedback loop, and just like your clot grows with a positive feedback loop, the mechanism that controls it also grows with a positive feedback loop. Which one wins out depends on which one is moving faster. In the early parts, while you have those platelets there pumping out all of their granules, you are going to have a clotting reaction that happens faster, all right? The farther away you get from that platelet plug, then the blood clotting reaction slows down, and this plasmin reaction takes over and starts to win out the farther away you get from the nucleus of the clot. In addition, over time, as cells are stimulated to grow and repair, the clotting reaction slows down and our dissolution reaction begins to win. This process of blood clot dissolution, taking apart the fibrin polymer, is also called fibrinolysis. Literally the cutting of fibrin. Now, like I said, clots can be very dangerous. They're essential. You gotta have them. If you don't have clots, you're in a very bad situation. But inappropriate clotting happening in the wrong place can be very, very bad for you. So your body has mechanisms to prevent inappropriate clotting. First off, remember how we said that platelets do not adhere to the prostacyclin-coated endothelium, right? This is called platelet repulsion. It stops platelets from initiating a clotting reaction on blood vessels that haven't been damaged. Second is thrombin dilution, right? So you make thrombin in the area of in the area of the injury, in the area of the clot. It's at very high concentrations there, and so it makes the clot grow very, very fast. But as blood moves into the area, that thrombin is going to get diluted out throughout the entire body, and then it will basically not work, right? It's at too low a concentration to work. This can be important because if, for instance, due to shock or injury, you have something that causes your blood pressure to go down, or your heart stops working, or your heart slows down, then your thrombin will not be diluted rapidly enough, and that can cause a blood clot to grow out of control. This is part of what happens in a heart attack, right? As the heart shuts down because it's not getting any blood, right? Then you're not having blood flowing, you're not having thrombin taken away, and the blood clot continues to grow inside of your coronary artery, or not your coronary artery, or one of your coronary arteries, and that's bad. That kills people. You also have throughout your blood natural anticoagulants, right? Things that your body makes to make sure that your blood does not clot in inappropriate places. There are several of them. Two I want to highlight here. First, it's heparin. Heparin is a very powerful anticoagulant. This is actually released by basophils and mast cells. You'll recognize basophils as being a white blood cell, and mast cells are kind of very related to to basophils, and are thought to derive from them, or possibly that they both derive from the same developmental cell. But mast cells are basically like basophils, except they're in your tissues rather than your blood. They initiate the inflammatory response, and that when they do so, they release the alarmone, histamine, and they also release heparin, right? Which the way heparin works is it interferes with prothrombin activator. Without prothrombin activator, you don't get prothrombin, and then you don't get fibrin. You don't make a clot. Heparin is also what is usually, it's one of the things that is used to prevent blood from coagulating when you take a blood sample, right? So you stick somebody with a needle and you take some blood out of them, well, you basically just triggered the extrinsic pathway. Why doesn't that blood clot? Well, it would, and it can, except you have to add anticoagulants to it. Usually if you're drawing blood out of a tube, into a vial or something like that, that tube is actually coated with heparin to prevent the blood from coagulating. And often there's heparin in the tube that you're drawing your blood into to prevent it from clotting. There are a few other anticoagulants that can be used. Citrate works pretty well. But heparin is sort of the industry standard here. There's also antithrombin. This is made in your body by your liver, and it basically just deactivates thrombin before it can activate on fibrinogen, or even afterwards. It just deactivates thrombin. That's why it's called antithrombin. So, too much or too little clotting can both be a problem for you. Let's take a look at some disorders that can happen. So first off, a lack of ability to clot is usually described as a hemophilia. Hemophilias are primarily genetic disorders. Although if you're on certain very strong drugs, you effectively become hemophiliac. Naturally, they only occur due to genetic means, right? There's three, I don't want to say common, but there's three well-known forms of genetic hemophilia. Two of them are sex-linked: hemophilia A and hemophilia B. Hemophilia A results in a missing factor 8, and hemophilia B results in a missing factor 9. Of them, A is the more common and B is less common. You'll note that both of these are in the intrinsic pathway, so they won't affect extrinsic clotting, like clotting from like a cut or an injury outside of the body, but they will affect intrinsic clotting. So anytime you do internal damage to a blood vessel, that just won't get fixed. So people who have hemophilia, like if they get a cut, that's not much of a problem. If they get a smallish cut, but if they get a bruise or some sort of internal bleeding, that's where it's really a problem. And in fact, physical exertion can cause pressure injuries on blood vessels. This is something that happens all the time. That's why you have an intrinsic pathway. Just the process of being alive does damage to your blood vessels. That damage just normally gets naturally healed. You've got a clot that forms, gets replaced, repaired, dissolutes. Without that intrinsic pathway, it causes just physical exertion, can cause internal bleeding, excruciating pain. The standard treatment for this is you've got to put clotting factors in there. So usually that is plasma transfusions. They don't have a problem with the amount of blood that they make. So it's not like an anemia where you need to replace the blood cells, but you need to give them plasma, because plasma has those clotting factors that they can't make. So transfusion with plasma or purified clotting factors. Factor 8, which is the missing factor in hemophilia A, can be made synthetically and produced by bacteria now. So now you can just inject factor 8 IV, and you basically you treat the hemophilia. It is it's a temporary thing, but it's a lot easier to get an IV injection of factor 8, once a month or however long it is. I don't know the schedule for it off the top of my head, than it is to get, plasma transfusions on a regular basis. So that's what happens if you lack clotting, lack the ability to clot. That's hemophilia. What if you clot when you shouldn't be clotting? All right? That's called a thrombosis. Comes from the term thrombus, which is a fancy word for a clot. Thrombosis is abnormal clotting in an unbroken vessel. There are several things that can cause thrombosis. They can be caused by, plaques on the inside of the blood vessel. You've got these cartilaginous and fatty plaques that can form, and then platelets can stick to those plaques because you've covered up that platelet repelling coating, and it can also occur in veins. If you're inactive, then your veins, blood will tend to pool in your veins, and if blood remains stagnant for too long, if it doesn't move, then it starts to clot. So if you are prone to clotting disorders, there are several factors that make you, several genetic factors that make you prone to clotting disorders. They will sometimes recommend that you not go on long airplane flights. And I actually had a friend who had a thrombosis, and he was advised by his doctor not to go on long airplane flights. And they asked me. They said, we're going on a short flight, but I think it goes up as high. Is it the air pressure? Is it the lack of oxygen? What causes this? No, it's not the air pressure. It's not the lack of oxygen. It doesn't have anything to do with it being an airplane flight. The thing is that if you're in an airplane, you're sitting down for a long time. You're not moving. That inactivation, that inactivity causes blood to pool in your veins, and if you have a tendency to form blood clots, that can cause a clot to start to form inside of those veins. It doesn't have anything to do with the altitude. It could happen just as easily on a bus or something like that, but usually with a bus, you're going to be getting up and walking around every so often, whereas on a plane, they tend to keep that fasten seatbelt sign on a lot more. But it's just the inactivity. You got to get up and walk around at least once an hour if you have a potential clotting disorder. Right? Usually tends to occur in the leg veins. Those are the largest veins, the farthest down, and the ones that are most likely to remain basically where blood is going to pool for the longest period of time. All right? A clot that lodges in a blood vessel and basically cuts off blood supply is called an embolus. It doesn't have to be a clot. It could be anything that cuts off blood vessel supply is called an embolus, but it is most frequently a clot. If a clot breaks free in your blood vessel, maybe you've got a thrombus in your leg vein, but then you get up and you start to move around and it gets into circulation, there are several places where it can lodge. Right? A pulmonary embolism is a clot that breaks free and travels into the lungs and lodges in the lungs, doing damage there. An infarction is what happens when it lodges in non-lung tissue. Your lungs have plenty of oxygen, they're lungs. But if it lodges in any sort of tissue that is a high oxygen requirement and it cuts off the oxygen supply because it plugs up the blood vessel, that causes an infarction, which causes tissue death. So, for instance, a myocardial infarction is any embolus that lodges in the pulmonary arteries and, not pulmonary arteries, sorry, in the coronary arteries and cuts off blood supply to part of the heart. If you have an infarction that cuts off blood supply to part of the brain, then that is a stroke. So how do we clotting disorders? Well, if you want to prevent clots, you have somebody who is prone to having clots happen, somebody who has a genetic factor or some other risk factor for it. There are several ways to prevent that. So vitamin K is required for making clotting factors. It's required by the liver for the liver to make the clotting factors. So coumarin or warfarin are both vitamin K antagonists that basically stop you from making clotting factors. If you're on warfarin for long enough, you basically become hemophiliac. It's a pretty I don't want to say nasty, but it's a pretty potent drug, right? Some people are on warfarin long term, but usually not high doses. There's also aspirin. Aspirin is much more mild. It suppresses thromboxane A2, which you'll recall is one of the things released from platelet granules. And so it is an anti-clotting drug, in addition to being a pain reliever. So anyone who's at risk of heart disease, heart attacks, is usually put on a once-a-day aspirin, and that just decreases the likelihood of forming clots. Remember, we said that heart attack is myocardial infarction. It's a clot that forms and blocks off a coronary artery. So aspirin reduces both the likelihood and the size of clots. There are other anticoagulants that are occasionally used, not so much as like constant medication, but Huraden from medicinal leeches has been used. Well, I mean, it says since 1884, but leeches have been used since forever, there's not a lot of circumstances where leeches are useful, but they actually are quite useful in some circumstances. Since leeches feed on blood, they would have to contain an anticoagulant, you think about it. Otherwise, the blood would clot inside of them. Vipers also contain in their venom a substance called arvin, which is also a potent anticoagulant. Those are very good at preventing clots that have that might form in the future. But what if you have a clot? You have a thrombosis or an embolism or something like that. How can you bust the clot? How can you actually make that clot go away? Well, there are several ways to do it. One is streptokinase. This is actually a protein that comes from bacteria, that bacteria use to they actually have two proteins called coagulase, which causes a blood clot, and streptokinase. They will actually make a clot around themselves, and they'll hide out inside of the clot away from your immune system where they can reproduce and get bigger, make a lot more of themselves. Then they'll make streptokinase, and that will bust the clot open, and then all the bacteria get into your blood. It's a pretty nasty reaction there, but that streptokinase enzyme has been isolated and can be used to dissolve clots. It's used to dissolve clots in coronary vessels. It digests almost any protein, so it can cause a lot of damage to the body if it's used. But it also works very potently and very quickly. And if you've got a clot in a coronary vessel, then we can worry about cleaning up the other damage later on. Right now, we have to get rid of that clot. There's also TPA. Remember, I talked about TPA. We said the TPA, tissue plasminogen activator, is going to turn plasminogen into plasmin, and the plasmin is going to dissolve, fibrinolyse that clot. Well, your body makes TPA at a particular level to help control the clotting reaction, but what if you wanted to bust a clot really fast? We actually can synthesize TPA in the lab, and you can just give somebody a local injection of TPA, and boom, you make a whole bunch of plasmin, and it starts chewing away at that clot. Also, hemantin, another leech protein, which can also bust through clots. All right, so that's what I have to tell you guys about the clotting mechanisms and the hemostasis mechanisms of the body. My next video will probably be on blood and circulatory system disorders. So look for that one coming up. Until then, study hard and pass those tests.